Hi, today we discuss immunoglobulins. Immunoglobulins. Immunoglobulins are gamma globulins, and they are tetramer. 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 This means there are 4 chains, 2 heavy chains, 2 light chain, and it is a shape of Y. Okay. So it is Y-shaped tetramer having 2 heavy chains and 2 light chains. Okay. In nutshell, I need to tell which are the immunoglobulins. So I always may say a mnemonic for it, that is called as GAMDE, or you can also say it as gamed. But if you remember GAMDE, it would be easier. So there is IgG, IgA, Ig stands for immunoglobulin, IgM, IgD and IgE. Why this sequence is important? Because in this sequence the concentration of immunoglobulin decreases, okay, in the body. So G is maximum, E is the least. So GAMDE, IgGAMDE. It is a Y-shaped tetramer having 2 heavy chains, 2 light chains. It is a gamma globulin. This much we have discussed. Now coming to how an Y-shaped tetramer of the gamma globulin looks. So it is Y-shaped. This means there is one chain over here, there is one chain over here, and there is one like this. So this is our Y. Now we are saying it is a tetramer. So this has to be a heavy chain. So this is a heavy chain. So one heavy chain, 2 heavy chains. This is a second heavy chain. And this has to be a light chain. So one light chain, second light chain. Because it is a light chain, we draw it with the light color. So 2 light chains, okay. So one heavy chain, second heavy chain, second light, one light chain, second light chain. So 2 light chains, 2 heavy chain. Now we have discussed this. We have formed the Y. Now Y is, these are joined by sulfide bonds. So this is the intra-chain sulfide bond. One heavy chain joining interchain. One heavy chain joining the second heavy chain. So this is the interchain sulfide bond. Okay. So this is called as an interchain sulfide bond. Interchain sulfide linkage or interchain disulfide linkage. Okay. But this is not all. There is an intra-chain disulfide linkage present everywhere. Over here. Here. You have to draw this in your exam also whenever it is asked. Immunoglobulins form a good short note, and immunoglobulins are important MCQ point of view also. So these are the intrachain sulfide bonds. Now we have drawn intrachain sulfide bond. Now what one is to be remembering is that where there is a carboxyl group, it is the constant region. Where there is a carboxyl group, it is a constant region. So carboxyl starts with C, and this is our constant region. This is constant H. And where there is amino terminal, it is variable H. Okay. So constant H, variable H. Similarly, constant L, variable L. Light chain. Constant L, variable L. There is one constant L, there is one variable L. But there is one variable H, heavy. But there are 3 constant H. So we number them. Constant H1, constant H2, constant H3. So we have discussed this also. So constant heavy chain 1, constant heavy chain 2, constant heavy chain 3. Okay. So we have discussed this. Constant light chain and variable light chain. This much is done. So there are 2 heavy chains. There are 2 light chains. There is an interchain disulfide linkage, there is an intra-chain disulfide linkage is present. This is the way the heavy chain is done. When there is carboxy terminal, it is a constant region, and when there is amino terminal, it is a variable region. Okay. Now, biochemistry without enzyme is not over any time. So there is an enzyme called as papain. Papain. When this acts, it acts on this region and it divides, separates the constant heavy chain 1 and constant heavy chain 2 and 3. So this is the one which is the upper region is called as fragment antigen binding. The one which is lower is called as fragment crystallizable. So fragment antigen binding, fragment crystallizable, since it just exactly cuts the disulfide linkages, separating the constant H1 from constant H2 and constant H3. This is called as the hinge region. This is called as a hinge region. Now we are done with the structure of immunoglobulin. It has a variable H chain, constant H1, constant H2, constant H3, carboxy terminal, amino terminal, many sulfide linkages present. Papain acts on the hinge region, separating it as fragment antigen binding, fragment crystallizable. Okay. So whenever this diagram is done, you have to draw this diagram very carefully and mention all the linkages. Now coming to the heavy chains and the light chains. We discussed that there is GAMD, IgG, GAMD, IgA, IgM, IgD and IgE. Okay, the concentration decreases this way. Second, the heavy chain, there is gamma heavy chain, G gamma, A alpha heavy chain, M mu heavy chain, D delta heavy chain, E epsilon heavy chain. Okay. 3. Light chains. Now light chains are of two types. It could be kappa or it could be lambda. Never both. If one has kappa, it will have both the kappas. If one has lambda, it will have both the lambdas. It is never both. It is never a mixture. Fourth. Now what is the importance of IgG, IgA, IgM, IgD, Ig? All are immunoglobulins. All will be helping in fighting infections. But how? First we will see, as I have told, we have to remember this sequence, and the concentration decreases this way. This means IgG is most abundant. IgG is most abundant. IgG is the least. IgE is the least. Okay. Now if we see MCQ point of view, there are many MCQs on IgG and IgE. Okay. So we will discuss it further. IgG. IgG is most abundant. IgG is most abundant. Most abundant. Second, it can cross the placenta. It can cross the placenta. It is having lowest carbohydrate. It is having lowest carbohydrate. It is having maximum half-life of 23 days. Okay, these are all MCQs. It is most abundant, it crosses the placenta, it has lowest carbohydrate, it has maximum half-life. Fine. Coming to IgE, it is increase in allergic conditions. It is increase in allergic conditions. It has highest carbohydrate. It has highest this IgE. IgE we are discussing. It has highest carbohydrate content. It has highest carbohydrate content. It is responsible for type 1 hypersensitivity. It is responsible for type 1 hypersensitivity. See, immunoglobulin is a topic which is present in biochemistry, which is present in microbiology, which is present in all the other subjects. It somehow comes. The MCQ will keep on repeating in whichever year you are or you are preparing for pre-PG. So it is increase in allergy, highest carbohydrate, type 1 hypersensitivity, and it is heat level. So IgG is most abundant, crosses placenta, lowest carbohydrate content, maximum T half. Ig is increase in allergic, highest carbohydrate content, type 1 hypersensitivity responsible, and heat level. So this is what we have seen till now. Coming to IgA. Okay, coming to IgA. Now what does IgA do? IgA is present in secretions. So it is present in body secretions. So it is present in all body secretions. Okay. And IgA can be present as Y, that is monomer, the diagram we saw, or it can be present as Y which is joined by a J chain. J chain. So it is a dimer joined by a J chain. Okay. This much we have to remember. And next important thing is that it is most importantly present in colostrum. Colostrum. Coming to IgM. IgM, it is largest. Now why it is largest of all immunoglobulin? Because it is not present as single Y, it is present as a flower shaped structure. Through a J chain. So it is actually a pentamer. There are 5 present. 5 monomers form a pentamer and there are 5 present. So and it is the first antibody to be produced in infection. To be produced in infection. So IgG, IgA, IgM, IgE. IgA is present in body secretions mostly and most important in colostrum. It can be a monomer, it can be a dimer through J chain. Okay. IgM is largest because it is pentameric structure joined through J chain and it is the first antibody to be produced in infections, and Ig is increased allergic condition, highest carbohydrate, type 1 hypersensitivity and heat level. In the whole discussion we left one, GAMD, we have left D. D is important. It has one important function, that it is present on B cells, on the surface of B cells. So IgD is present on surface of B cells. So this we have to remember. Other things also in the immunoglobulins we have to remember: Ig, gamma, IgA, IgA, IgM, IgD, IgE. The concentration decreases. The carb content is decreased over here, it is increased over here. The T-half is max over here. This is heat level. This is present in secretions. This is largest and first antibody. Largest and first antibody, and this is on B cell. To summarize, okay, that's all for immunoglobulins. Thank you very much.